Hi, thank you so much for coming to my presentation. My name is Khera. I'm 14 years old and I'm really passionate about human longevity. Today I'm going to be talking about human longevity, current research and barriers, and my research in the field. As you can see, that is my own drawing. And I drew it just because, well, I want to talk to you about having a relative that, is going through an aging-related disease. If you had a relative that was going through an aging-related disease like dementia, Parkinson's, or any of that, a cardiovascular disease, anything past the age of 50, then that is likely an aging-related disease, and it's really unfortunate. And there are a few things, causing it and causing this sort of decline in terms of memory, in terms of health, in terms of overall fatigue. And pretty much, this is actually way more common than you think. And for dementia, 1 in 6 women and 1 in 10 men who live past the age of 65 will get dementia at some point in their lifetime. That's pretty shocking and scary, right? Well, the thing is, dementia is in part caused by, we're going to use this as dementia as sort of a case, but dementia is actually in part caused by aging. It's furthered by aging because it's an age-related disease. And aging can sort of be defined as the process of going old, losing muscle mass, getting tired, progressively, getting gray hair. And there's a lot of different, causes, and there's a lot of different phenotypes, or kind of manifestations of aging. And I'm going to talk about a few of the hallmarks, so the main causes of aging today. I'm not going to talk about all of them because there are a lot, and we're discovering new ones by the second. But I'm going to talk about really important ones. So first, I'm going to talk about cellular senescence. So cellular senescence can sort of be described as the zombification of a cell. When your cell becomes a little bit bigger and a little bit more messed up inside networks, and also is releasing sort of a protein, an associated protein that makes other cells turn senescent as well, which is not good. And it's almost like a zombie apocalypse happening to your body, progressively getting faster because of the higher accumulation of these cells. And it's typically caused by a few things. First, I'm going to talk about telomeres. As you can see, or as you may not see, those are aglets or shoelaces. Oh, well, those are shoelaces with aglets on them. And pretty much, you can also define them as telomeres. So telomeres are pretty much, well, when your cell divides, the DNA is copied from chromosome to chromosome. And unfortunately, this process has a small error, and DNA is cut off at the ends of your chromosomes. And that's not good, because pretty much that means that you're going to lose a bunch of valuable DNA information. So your body has these things called telomeres, which are redundant pieces of DNA at the ends of your chromosomes can be cut off. And after about 50 to 70 divisions, telomeres are cut off and your cells become senescent. There are also some other causes, like oncogenic signals. So when a cell is about to become cancerous, since senescence is pretty much the opposite of cancer. Senescence is basically when a cell becomes zombie-like, and cancer is basically when a cell won't stop dividing. There's also metagenic signals. So signals from cells at several points in their cycle. Undefined stress, so different stressors posing on it, the cell, and also DNA damage. So this is actually what I'm going to talk about in a second. But having problems with your genetic code, so hurting the genetic code that powers us. And pretty much DNA damage can be defined in terms of a few things. This is sort of, as you can see, or maybe not see, a strand of DNA. And typically when there's radiation or different stressors that can actually cause mutations and cause breaks in your strands of DNA, switching out different letters in your DNA code for ones that don't really work. That's not very good. And that actually, coincides in it does cause aging. And it causes a lot of these other causes of aging. And also there's epigenetics. So this can sort of be defined as the way the DNA is stored in the cell. So your DNA is stored in your cell's information center, like the city center of a different city, called a nucleus. And the way it stores is it's wrapped around these proteins called histones. As you can see, I drew this too. And pretty much the histones are proteins that they hold the DNA, the genes. And so when DNA is firmly wrapped around a histone, it's typically not expressed because it's hard to get to that DNA and transcribe it so you can actually make the products that come from that DNA. But when DNA is out in the open, it is expressed. So based on what genes are in the DNA are, wrapped around the histones, those genes that make you up, depending on which are being expressed, you'll exhibit more of that certain factor. So as you age, there are these things called epigenetic alterations. And that's when your DNA and you well, pretty much your DNA. So this is the epigenome, as you can see my epigene board, and pretty much your epigenetics. So there are different chemical tags that get put on your. This is sort of a general, descriptive page poster, and here's sort of making it a bit more simple. But pretty much chemical tags dictate whether DNA is wrapped around said histones and whether it's expressed or not. So pink ones in this case might mean that it might be wrapped around a histone, and the blue ones in this case might mean it's not. So yeah, this basically dictates whether they're expressed or not. And as you age, there are little alterations in this that can either be good for you, like the expression of a gene that is objectively good, or bad. So the expression of a gene that does not do you that much good, or the lost expression of a gene that is good for you. Then there's also gut bacteria. So, I mean, I'm sure you guys have all heard of bacteria, especially with the ongoing pandemic. I know it's a virus, but there's also, we get very concerned about bacteria and what's kind of resting on surfaces. Did you know we actually have more bacteria than we have cells in our body? That's wow. And it's a lot more bacteria. So pretty much this bacteria is sort of situated in your stomach. And this is actually typically good for us. So this is part of our microbiome, or our gut microbiome in this case. So pretty much we can look at these types of bacteria as Gabe, the good bacterium, and Bob, the bad bacterium. And some bacteria are not so great for us. And they're pathogenic, they're not great. But some are good. And pretty much there's also, dysbiosis. So where there's different balances of bacteria. So there can be more of a certain bacteria, and you might not want to have more of that certain bacteria. Now, so someone with an aging-related disease might have more of the bad bacteria, or more of a certain type of bacteria you don't want to have a lot of. It might do good in small quantities, but not great in a lot of quantities. And then also, someone without an aging-related disease might have more good bacteria, or a better sort of overall sense of health in their microbiome, better biosis. In this case, it might be dysbiosis. So then there's oxidative stress. So this is sort of when molecules play a cyclical and mean game of tag. So there's these things on molecules, or electrons. In molecules, there's these things called valence shells. So that's where there are electrons on the ends of the molecules, and pretty much, or the atoms, and pretty much, you want about 8. You want about 8 if you're well, I don't want to explain the entire thing, but typically, molecules want about 8 electrons in their valence shell, because that makes a complete valence shell. So when molecules have 7, or number that's a little bit less than that, they might want to steal electrons from another molecule, which definitely isn't good, because it does this thing called oxidation. That's almost when an apple turns brown. That's what happens. Different molecules are sort of attacking and playing tag with each other. So you can kind of see they're called free radicals here. So these are the black free radicals, and and obviously there's more molecules than this in one's cell, but pretty much here's what you kind of see for now. I'm focusing on these, but they can come closer, and they can actually turn these molecules into free radicals, and they can have their own electrons, which causes oxidation. And then NAD plus also is a little bit beneficial in this sense, because it's this other molecule that can pretty much act as an electron donor. So while with not hurting itself, it could actually give, it could give electrons to other molecules. So it could look a little bit like this, which is great. So, and now I've kind of gone over a few hallmarks. There are other ones like mitochondria deterioration. So those are the powerhouse of your cells. There's also neuronal aging and brain aging. So, the deterioration of what makes us up, neuroscience. And so implications are sort of almost the unfortunate aftermath of aging. And there are a few implications here. I mean, aging, I'm going to list that as an implication because there are a lot of, ways that aging is sort of exemplified in our bodies. But then there's also health span. So the amount of your life that you spend healthy, that kind of deteriorating and that getting way smaller if we kind of succumb to the effects of aging. There's also neurodegeneration. So again, neuronal aging. So actually watching those brain cells sort of die off due to potentially lack of oxygen, tricky blood vessels. What happens with vascular dementia is that blood vessels going to the brain, because your brain cells need oxygen constantly. So, blood vessels going to the brain actually get leaky, or they get blocked, and then your brain can't get oxygen fast enough. And so then there's also lifespan, so a lower lifespan, diseases, different diseases like Parkinson's, again, dementia, different cardiovascular diseases. Then there's also, solutions. Because I mean, I've been a little bit cynical so far. Like, what are actually ways we're looking at to prevent this unfortunate aftermath? So that is right now intermittent fasting. So, well, that's one of them. It's sort of a fad diet you probably heard of. And what it does is it initiates this process called autophagy. I mean, what it is fasting about 16 hours a day and eating for about 8, or fasting 24 hours twice a week. And what this does, or is theorized to do, and we're kind of investigating the mechanisms right now, is actually look at a process called autophagy. And autophagy, I would call in short, it's sort of a process where cells are almost recycled. So senescent cells, so damaged cells, are sort of eaten as fuel, which is sort of your body kind of basically eating itself, but in a good way. Obviously, going too far could hurt your healthy cells. So I don't recommend you do this yet because there's research contradicting those facts, and it's not fully safe just yet. I don't recommend, except nutrition and exercise, probably, and meditation. I don't recommend doing pharmaceuticals or intermittent fasting because they aren't fully proven. So there's obviously nutrition, so eating the right amount of food, the right diet, because that definitely aids your cells and their function because it's all for your body, right? There's exercise. So exercise is actually found to slow the rate of premature death and basically act like medicine for older adults. Also meditation. So just taking time out of your day not to exercise your neurons too much, because basically your neurons get tired over time, which is what causes their aging. And then there's pharmaceuticals. So there's a lot of different drugs being tested right now to eradicate these factors I told you about earlier, these hallmarks. And a lot of them are proven to be very effective, but obviously they're only in phase 1, phase 2, very early stage clinical trials. And so I was going to talk a little bit about the barriers that are kind of in our way. And pretty much there are 3 main ones that I'm kind of bringing it down to. There's FDA approval. So not really FDA approval for drugs so much as their acknowledgement of aging as a disease, aging as something that we should treat. And obviously there are some difficulties with running clinical studies around this. I don't think anyone wants to run a 70-year or 80-year clinical study. So that's why people focus on aging-related diseases for now. So seeing, hey, how can my drug target this one disease? And so, the FDA, unfortunately, doesn't classify aging as a disease, so we can't test it in even small scale. So mice, this trial on mice. I mean, there have been trials just for general experiments, but, FDA-controlled trials are, very scarce in terms of aging. And then there's also public opinion. So having that opinion about aging as a disease, raising awareness about that is really important because, the public controls what drugs you put to market, really everything, because it's the public that's actually gonna let you market that, because a lot of it's based on public approval. And then there's also funding. So the money kind of being put towards your work, the money being put towards your towards aging as a disease, it isn't as high. I mean, it's definitely increasing, but it isn't as high as it probably should be. So definitely increasing funding in that area would be a great thing to do, because then you could run more experiments and make more awesome discoveries. And so actually, I decided now I'm going to talk a little bit about my own experiments and, the research I'm doing. So pretty much I'm working with senescent cell mitochondria. So I mentioned senescent cells earlier, and they're like zombie type cells again. So the mitochondria are the powerhouse of your cell, and the mitochondria in senescent cells degrades a lot. There are a lot of papers supporting this hypothesis that, mitochondria in senescent cells are not doing well. And pretty much, I had an idea a little while ago. Actually, originally the idea was to look at senescent cells as a total and kind of distinguish between them using different aspects of machine learning, which I will explain later. But with a lot of guidance and mentorship, I narrowed it down to mitochondria, because focusing on a specific organelle might work a little bit better in terms of specificity. So yeah, this is sort of what mitochondria look a little bit like. They're just sort of oval, circular, and they have sort of different networks inside them. I wasn't going to draw the networks for you because it gets really complicated. And so pretty much what happens over time is the cells sort of get messed up. Like the networks inside go from being kind of straight to really, really messed up, which is not what we want for sure. And they're just sort of messing up the mitochondrial network. The cells get a bit bigger. Actually, sometimes they can double in size, which is a big size for cells, which actually means that they take up less room for healthy cells. But yeah, so the mitochondria, and actually I'm using AI to look at this. So I'm going to explain a little bit of a concept within AI to you today. So we're looking at machine learning. So machine learning is pretty much where you can teach a machine to do something, and it likely does it better than humans because we're a bit biased, and obviously there's the issue of the code being biased, but this is definitely a step up. So we're actually looking at something called a convolutional neural network, or a CNN, as you can see there. So there are a few layers to this convolutional neural network, and I'm going to walk you through this process overall. So first, it takes in the input of cells or mitochondria or whatever whatever you want to put in there. Then, first the input, and then it takes you to the convolutional layer. So this is where, pretty much it looks at all the features and it kind of creates a general combination of them in a way. So it picks all the features, it kind of compresses them together. Then in the max pooling layer, it sort of does the same thing, but it picks the most prominent features. And then from there to the classification layer, so it basically says, hey, so I noticed that these cells are senescent, these cells are not senescent. So this is proliferating, so P means proliferating. So proliferating is sort of normally dividing, and that's senescent. So it basically categorizes them based off of the remaining numbers. It goes through this about 2 times. So then from there, basically I've been working on this sort of problem with a company called BioTeam. And this is a company I founded about a year ago. And we're basically focusing on you're running this algorithm to find phenotypes of senescent cells, so key features of senescent cells. So we translated into biomarkers or different processes we can infer that the senescent cells have afterwards. And we're working on a bunch of, other than convolutional neural network, we're working on a bunch of different analyses otherwise. So there's autoencoders too. There's also just different image segmentation. So autoencoders are another type of machine learning model where you pretty much you just shrink the image down to its key features. It's not really about classification. And then we're also working with image segmentation, so just running the images through kind of something that will profile them, say, hey, this is, the length of the cell is this much, the length of this mitochondria is this much versus the other length of the proliferating mitochondria. And so we have a vision to eradicate aging and aging-related diseases, and that's sort of what's really important to us. So we think, with these approaches, with everything that we're doing now in the aging field, we might be able to kind of go from someone who's having dementia or having these sort of symptoms to someone who's actually fully cured and is happy with their family and is living a good life. Because really, dementia and these other diseases don't just impact the patients, but they also impact the family and the people around them. So this is a little bit about me. And that's my website, and that's my company's website, and thank you so much. I think this is pretty much it. So I have a few questions for me. And so first is, do you have any pieces of advice for aspiring young researchers? So I honestly say to study the field you're interested in a lot. Read papers, prepare, understand it really well, understand all the current literature, or subscribe to those papers. There's a really good one called Meta, where you basically can subscribe to it and basically get sort of a digest every week of different papers that are in the field that you choose. So that's really interesting, just reading all the current news in the field. Also speak to those in the field to get a good understanding. Do I love this field? What is it like working here? And also, take note of the ideas you have with the possibility of pushing them forward. I think that's amazing. What have been the things you learned along your journey? So something I've definitely learned a lot. I've learned a lot during this journey, and one of the main things is just to sort of take initiative, do it. Don't psych yourself out. Don't be scared. And I used to get scared about sending emails or talking to people, but now I sort of remind myself that I won't succeed at anything I don't do. I think it's by Michael Jordan. The quote that you'll miss all the shots you don't take, and so to speak. So I've also learned that habits sort of make a person. So the compounding of small habits, small things in your day-to-day life that you have control over and that you help make better, is an amazing thing to do. And so so the small things sort of make out the big. The person who you are, you can choose that. You can choose it that way. And also, how can data privacy issues affect the further development of genomics? That's a really interesting question. So there's obviously a lot of squabbles about data privacy nowadays because having different studies with genomics means that you might know things about the entire person, and it's a little bit worrying. So one thing that I definitely think that could help this or could stop this is not releasing any personal information. Obviously, race, gender, that might, need to be added, but, person name, different things when conducting these studies. That would be really important, just not to release that to the researchers. Of course, it could affect the further development because just in general, if we have data privacy issues, people might be concerned about sending data in. They might be concerned about, participating in these studies. So that might not be good. And yeah, one more is, what has your journey been in human longevity? So medicine and coding have always been continuous passions of mine. And I learned about human longevity in late 2018. From there, I read a bunch of papers and learned a lot more. When in March, I sort of came up with an idea the idea I just mentioned to you, to use machine learning and basically find these phenotypes or these key features of these senescent zombie type cells in your body, furthering aging-related diseases. And sort of one thing led to another, and now I got a lot deeper into longevity, and I'm tackling a bunch of different hallmarks of aging. This is something I'm really passionate about. But yeah, I think that's sort of my overall journey. Oh, and yeah, one more. So what got you interested in longevity in the first place, and how old were you? So, I was about 12, I'd say, when I got interested. And what got me interested? Well, I don't know. It was just, I did a lot of reading. There were a lot of, different organizations that provided support. So I signed up for some different programs. And yeah, so just kind of doing a lot of reading, different papers, reading them, it just got me really fascinated. And yeah, I think that's sort of like what got me interested. Another thing that got me interested, just seeing relatives, go through different aging-related diseases. That's always, really saddened me, but also it's something that, I want to solve and I want to, change as a result. So I think that definitely was what got me interested in. Yeah. I don't think there are any more questions, but if there are, you can contact me likely on my website, at ninakhera.com. Contact me there. And yeah, there. And yeah, I think that's pretty much it. Thank you so much for having me, and I hope you enjoyed my talk.